Good evening, everyone, and welcome to the University of Saskatchewan's 2013 Whelen Lecture. My name is Patty McDougall, and I'm the Vice Provost of Teaching and Learning at the University of Saskatchewan. I'm standing in for our Provost and Vice President Academic, Brett Fairbairn, this evening at this evening's lecture, and it's my pleasure to do so. As Vice Provost Teaching and Learning, the very best part of my job is being able to focus my attention, my energies on strategic innovations in our academic programs. It was my privilege today to spend time with tonight's lecturer as we traveled around campus, spending time with faculty and graduate and undergraduate students and watching him share his innovative work so generously with our campus. Let me tell you a little bit about tonight's event. The Whelen Lecture Series was originally established in 1986 at the bequest of Dr. Myron Whelen, a distinguished alumnus from the University of Saskatchewan. And Dr. Whelen came from the small farming community of Birch Hills in northeastern Saskatchewan, recognizing that communities in our province often lack opportunities to hear and learn from internationally recognized people. The mandate of his legacy, the Whelen Lecture, is to bring to this campus, to this city, an internationally recognized authority in a discipline taught at this university. I suspect Dr. Whelen would be very pleased that we're streaming this lecture so that others beyond those in the room will be able to watch it and to be part of it. In keeping with the spirit of Dr. Whelen's bequest and in the tradition of the University of Saskatchewan, the program for the Whelen Visiting Lectureship is designed for a broad spectrum of people: students, faculty, researchers, teachers, professionals, public interest groups, and private citizens. Visiting lecturers have an international reputation within their fields, and this year's speaker is no exception, as you're about to see. I'm going to hand over the job tonight of introducing our esteemed lecturer to my colleague, Dr. Daniel Chen, who is a researcher and professor in the Department of Mechanical Engineering and the Division of Biomedical Engineering here at the University of Saskatchewan. Dr. Chen is leading our own tissue engineering research that spans both engineering and life sciences, and Daniel is developing advanced technologies for the production of various scaffold-guided tissue and organ substitutes. Please welcome Dr. Daniel Chen.
Thank you and good evening. And over the last decade, the demand for organ transplants has doubled. And yet, the availability of viable organs has remained fixed. Today, we have a severe organ crisis and a rapidly growing number of patients on the transplanting waiting list who are dying. Imagine having the ability of regenerate living and functional tissues that can repair and replace the damaged organs. Imagine the possibility in growing these tissues and organs in the lab, and then, when ready, safely transplanting them in our patients. Imagine a day that there's no longer organ shortage crisis and countless lives to be saved. This is the dawn of that day. So it's my honor to introduce to tonight's speaker, a groundbreaking research in regenerative medicine, Dr. Anthony Atana. Dr. Anthony Atana is the director of Rick Forest Institute for Regenerative Medicine and a W.H. Boyce Professor, and the chair of the Department of Urology at the Rick Forest University. He is a practicing surgeon and a leading authority in the area of regenerative medicine. His current work focuses on the growing new human cells, tissues, and organs. Dr. Adler has received numerous awards, which include the U.S. Congress-funded Christopher Columbus Foundation Award that is bestowed on the living American who is currently working on discovery that will significantly affect society. He has received the World Technology Award in Health and Medicine, which is presented to those who have achieved a significant and lasting progress. He was also awarded the Seminole Gross Prize given every 5 years to a national leading surgical researcher by the Philadelphia Academy of Surgery. In 2011, he was elected to the Institute of Medicine of National Academy of Sciences. The place has also recognized Dr. Adler's achievement, and in 2003, Scientific American named him as the Medical Treatment Leader of the Year for his contribution on the field of cells, tissues, and organ regeneration. His work was listed as Discover Magazine's #1 top science story of the year in the field of medicine in 2007. That same year, a Time magazine poll ranked him as the 56th most influential person. In 2009, Dr. Adler was featured in the U.S. News and World Report as one of the 14 pioneers of medicine progress in 21st century. Dr. Adler's work was also listed in the Huffington Post as one of 18 great ideas of 2011, and in Time magazine as one of the top medical breakthroughs of the year. Dr. Adler has led and served on several professional and government committees, which include National Institutes of Health Working Group on Cells and Development Biology, the National Institute of Health Bioengineering Consortium, and the National Cancer Institute's Advisory Board. He is the editor of 12 books, including The Principle of Regenerative Medicine and the Methods of Tissue Engineering. He has published more than 400 journal articles and has applied or received over 200 national and international patents. So please join me in extending a warm welcome to our distinguished speaker, Dr. Ashley Adler.
Thank you, Dan. Thank you so much. Appreciate it. Thank you, Dan, for that very nice introduction. It's really nice to be with all of you today. I've just had a fabulous day today. I've been at the institution all day visiting your beautiful city of Saskatoon. What an amazing place you have here, and what a gem you have with the university. It's just been really a treat. I was greeted this morning by Karen Hayward, and I just had a wonderful time going from different sessions with students. I had the pleasure of interacting with Patty McDougall, who you heard of earlier today as the Vice Provost. You now have heard my sessions all day long, so I wonder what you're going to probably fall asleep tonight. And then I had the wonderful pleasure of having a session with the scientists and the researchers, and then a nice luncheon at the University Club. Bob Cram, thank you so much for hosting that event. I've had just a wonderful time there. And also Hope Belinsky with the nursing school, who actually hosted my session with the nursing students. And last but not least, I would like to thank Vanessa Young, who actually has really made all the arrangements for me to be here today. So thanks all of you for having me. Thank you for hosting me. What I thought I would do today is really give you an overview of some of the work that's being done in the area of regenerative medicine. And this is actually, we're going to start here. Let's see. Can you see the slides? There they are. Okay. Very good. So this is actually the institute that the Wake Forest Institute for Regenerative Medicine that you see there in the front. And what I'm going to do is talk to you a little bit about the history of regenerative medicine. Then I'll talk to you a little bit about some of the challenges and principles that we have followed, give you some examples of the field, and show you where we're going for the future. This is actually a painting that hangs at the Countway Library at Harvard Medical School, and it shows the very first time an organ was ever transplanted. And in the back room, you see Hartwell Harrison, who was the chief of urology at Harvard, and he's retrieving the organ, which happened to be a kidney. And in the front room, you see Joe Murray. Joe was a plastic surgeon and had worked his whole life trying to come up with a system to transplant organs. and Joe Mary's getting the patient ready for the transplant, and that was back in 1954. This was the very first time that an organ was ever transplanted, and so many lives that have been saved by this one single accomplishment. Joe Mary went on to win the Nobel Prize for this work, and so many lives that were saved, but still so many lives that we can save today due to a major organ shortage. In fact, the statistics are fairly significant. Every 30 seconds, a patient dies from diseases that could be treated with donor tissue or organs. Now, wouldn't it be great if we could regenerate ourselves? Well, actually, it's not unusual. There are many things in nature that do regenerate themselves. This is a salamander, and you can see the salamander limb, and this is an injured limb that totally was regrown in a couple of weeks. The salamander just did it on its own. So if a salamander can do it, why can't we? Why can't humans? Well, the fact is that we do have the potential to regenerate, and you're regenerating all the time. You're regenerating every single day. Every 10 days, your skin is turning over. Every week or so, the lining in your intestine is turning over. Your bone takes about 10 years for your bones to fully be replaced. It takes about 20 years for for all your brain cells to be replaced, which explains the behavior of some of our teenagers. But actually, we are constantly regenerating. And that's really what allows us to follow this field that we call regenerative medicine. And it's not really a new field. It really is based on the field of cell transplantation. And this dates back to the 1930s. This is a textbook published back in 1938 titled The Culture of Organs. And you can see the authors there. The first author is Alexis Carrel. And Alexis, like Joe Mary, was another Nobel Prize winner. He actually was a French surgeon from Lyon, France. And his mother was a seamstress. And he took a piece of hosiery material and he sterilized it in alcohol and used it as a vascular graft. It was the very first time that a vascular graft had ever been implanted in a patient. And he also devised some of the suturing techniques we still use today in surgery. His co-author in the book was Charles Lindbergh. That is the same Charles Lindbergh who actually flew across the Atlantic back in the 1920s. He spent the rest of his life working at the Rockefeller Institute in New York in the area of the culture of organs. The very first time that tissues were ever transplanted in a patient or cells were ever used for regeneration dates back to 1981. In that year, a patient at the Mass General Hospital in Boston with a burn had a very small piece of skin taken, less than half the size of a postage stamp. They expanded the skin cells outside the body, normal cells. They placed the cells on a piece of gauze, a dressing, and placed the cells over the burn area. Now, it didn't generate a new skin segment, but it did show that the wound healing was accelerated. And it was the very first time that the patient's own cells were actually used for regenerative therapy. That was back in 1981. So if the field's been around for so long, why so few clinical advances? And this really has to do with several challenges in the field. And if I were to point to three major challenges that we saw in the field, they're listed right here. The first one was an inherent inability to grow cells outside the body in large quantities. The thought was that it just couldn't be done. You couldn't take cells from a patient and allow those cells to grow and expand. Couldn't be done. That was the first challenge. The second challenge was inadequate materials. Even if you are able to grow cells, how can you deliver cells back into the body if you want to use them for treatment? And the third challenge was inadequate blood supply. Even if we wanted to place these cells inside the body, the cells had a major challenge of surviving inside the body once again if you put them back in the body without a blood vessel supply. So this is what really got us into the field. Many years ago, we started working on growth factor biology, how we could actually get these cells to grow outside the body. How could we take cells from your own tissues and organs and allow those cells to grow and expand? And basically, by looking at the growth factor biology, and looking at where these cells were located, we were now able to take these cells that before could not be grown. We could now take a very small piece of tissue, less than half the size of a postage stamp, and by day 60, we could have enough cells to cover a football field. And this was done just by knowing the right growth factors and where these cells were located. And we started doing that for a few cell types. And now, if we fast forward about 25 years later, basically many scientists that have made major advances in the ability of growing cells outside the body. So if you were to look at this list right now, all of these cell types right now can be grown outside the body, all the ones listed in white. But if you were to look at the ones listed in black, and I don't know if you can see them clearly, but those are liver, pancreas, and nerve. Even now, 2013, with all the advances we have made in the field of science, these cells still cannot be grown from a patient. That is, we cannot go to a patient, take a small piece of their tissue from the pancreas, the liver, or the nerve, and we cannot grow those cells outside the body. So there's still advances that need to be made. The second challenge was, how do we get these cells inside the body? How do we deliver the cells? And basically, we needed to deliver the cells using safe materials, materials that we could get into the body and they would allow the cells to survive. And the third challenge was the blood supply. We were very lucky that back in our early days of research, our floor mate in our research building was the founder of this field called angiogenesis. And he started working on this field of how do blood vessels really grow. And he, back in 1973, established this very simple principle, and that is that cells inside the body could not grow or be expanded if you implanted them in volumes larger than 3 millimeters cubed. Now, this was a major challenge because I just mentioned to you that we can now grow enough cells to cover a football field. But if we want to use those cells in a patient, we can only inject cells about the size of a pencil eraser. Otherwise, they will not survive because they don't have a blood supply. So how does nature solve this problem? Nature solves this problem by branching. From the lowest forms of life, such as algae, which you may see at the bottom of the ocean floor, to an individual leaf coming from a plant, or the branches that you see in a tree, or the blood vessel branches you see in a lung, or the branches you see in all of the human body. Branching is a solution to nutrient diffusion. And that is why, basically, many scientists, biomedical engineers and material scientists, started to design scaffold systems, materials that we could put inside the body. But these materials look like a piece of your shirt or your blouse to the naked eye, like you see on the left. But under the microscope, on the right panel, you see this branching pattern, these fibers that under the microscope, you have these branching patterns. It looks like a piece of your blouse or your shirt, but on the microscope, it has this branching pattern which allows the cells to be delivered inside the body, and it allows the blood vessels to supply this because you have the pores present, and those pores, that branching, allows the blood vessels in your body to feed the cells that we put in. So basically, when we talk about regenerative medicine, those are some of the basic scientific principles for some of the students here tonight. And but now we go into the real field of regenerative medicine. How do we actually treat these patients? And we can treat these patients by either using cells alone, using scaffolds alone, which are these materials I just showed you, or using cells and materials together. Now, I'm going to show you first how we use just materials alone to regenerate organs. Basically, what you see here in this video is you're seeing this video of, looks like a cotton candy machine. But instead of sugar, you're seeing these fibers that are going around this mandrel. And this mandrel then forms this tubularized structure that you see here. And these materials that we create in the laboratory totally dissolve on their own once inside the body. And we can make them in different sizes and different shapes. So here's a patient who presented to us with an injury. this is an X-ray on the top. And you can see on the X-ray, that you have a normal organ on the right, a normal organ on the left, but in the central portion, you can see that the organ looks a little bit injured. It looks narrowed, strictured. You can see the stricture right in the center of that X-ray. Well, now what we wanted to do is we wanted to replace this defect using just the material alone without the cells, just this scaffold that we created. That scaffold that I showed you that looks like a piece of your blouse or your shirt, but under the microscope it has this branching pattern. But we're going to use it without cells. And the way that we do that is as follows. What we did is we took these materials that we created and we then replaced only the top portion of the injured organ. So if you notice in that X-ray, the bottom portion actually is still normal. It's only the top portion which has the defect. So what we ended up doing is we just replaced the top portion. We removed the injured tissue and we replaced that with this material that you see here in the center. And by doing so, we were able to replace the entire top length of that defective area. And then what happened was that in this next slide, you're going to see what actually happened. There's the X-ray on the far left of the injured organ, and there's the X-ray on the bottom right of the regenerated organ. And that now is fully regenerated. That's an X-ray, but when we take a small piece of that tissue, it's fully regenerated. So what's happened here? How did that happen? Well, basically, your body regenerated itself. That's what happened. And it used that scaffold as a bridge. So we used that scaffold as the bridge, and the cells from the bottom used that bridge to walk on that bridge. The cells walked on that bridge, and they bridged the gap. And then it regenerated the organ fully. And that's what happened in that patient. And that happens because you can regenerate, but you can regenerate for limited distances only. And that only works for small distances, about 0.5 centimeter from any edge. So it only works for small defects. So here's the next technology that we used was using cells alone. So that technology is using materials alone to regenerate your body, and that works for small distances. For larger distances, you do need cells. And so the way that we do that is we use cells for many different types of therapies, many different types of treatments. But I'm going to give you one example right here. This is actually a picture of skin. It's just a drawing of what your skin looks like. But if you were to have a burn, you would be damaging part of your skin. You would have a burn. So the way that we're using cell therapy, for example, is in this next slide. We bring the patient into the operating room, and we have a kit that you can see here in the top. You can see that kit on the top, and a small piece of skin is taken from the patient, very small piece of skin. We're able to take the cells from that piece of skin, and we're able to lay those cells on the patient and spray them on the spray like you saw on the slide. The cells are actually sprayed onto the burn area, and they cover the burn, and the patients now have new skin. And it's just a spray of skin. And this is technology that has been led in the U.S. and in 10 centers around the U.S. by Jimmy Holmes from Wake Forest. He's one of our burn surgeons, and it's technology that's currently being used clinically. The next type of strategy is to actually use cells and materials together. And the way that works is we use the same type of materials I just showed you, and we use the same kind of cells from the patients, but we just put them together. This is an example. We go to the patient, and let's say the patient has an injured organ or tissue. So we take a very small piece of that tissue from the patient. In this case, I'm showing you a piece of muscle. We take a small piece of the muscle. We then tease the cells apart, and those cells grow on their own. So we put them in culture plates where they start growing on their own. We start expanding the cells outside the body. And then a few weeks later, we have enough cells, and we then take that scaffold, that material I showed you, that looks like a piece of your shirt or your blouse, and we put the cells over the material. That's what we do. And then for this piece of muscle, we then use these exercise machines that actually exercise these muscle strips. This is what we call a bioreactor, and it's exercising these muscle strips that we're creating in the laboratory so that they know how to do, and they're strong enough to do what they need to do once we implant them in the body. So that's an example of how we use cells and materials together. In this next slide, I'm going to show you another patient who presented to us with a much more severe injury. This is a severe injury in this patient, and you can see here the major area of the defect. Here you can see this major gap in this organ right here. You can see that's a very large defect in this patient, very large defect. This patient was in a car accident. It shattered the organ, so there was no organ left in that segment. So there was no way that we could use the material alone, right? This is a much larger defect. I mentioned to you those materials only work for small distances. So what did we do here? Well, here the strategy then was to take a very small piece of tissue from that same organ. We took a very small piece of tissue, less than half the size of a postage stamp. We then teased the cells apart from that piece of tissue. That organ's made up of two different cell types, so we grew the cells separately. We then took those cells and we then coated the outside. We took that scaffold, we tubularized the material, and we then coated those muscle cells on the outside, and we coated the lining cells that are supposed to be in that organ on the inside. It's very much like baking a layer cake, if you will. Okay, you're doing it one layer at a time. Or another analogy is you're making a peanut butter and jelly sandwich. Okay, you're just taking the piece of bread, you're putting jelly, and then you're putting your peanut butter, then the jelly, and that's exactly what you're doing here. You're placing these layers of cells on these scaffolds, and then you place this structure in this oven-like device that has the same conditions as the human body. You let it cook there for a while, okay, same conditions as the human body, these oven-like devices, and then you put it back into the patient, as you see in the diagram. So this is actually a slide of the patient's x-ray on the left with a defect. On the second panel, you see the scaffold that we tubularized with the scaffold alone. In the third panel, you see a drawing of one of these bioreactors where we actually allow the structure to cook, if you will. On the far right, you see the structure now fully coated with cells. It's now ready to be implanted. And when you look at this next slide, you're going to look at the x-ray on the left, which is before surgery. And in the center, you're seeing the x-ray after the surgery, showing you now the normal organ that was present there. And we published that work in 2006 in this journal called The Lancet. And at that point, when we published a series, we had a series of patients with up to a 6-year follow-up. We followed them for 6 years before we published that paper, showing how that technology works. Now, we've followed the same strategy for blood vessels. This is actually a blood vessel that we have in a bioreactor. You can see it pumping away. And the way that this works, same strategy. We take these materials that look like a piece of your shirt or your blouse. We tubularize them. A blood vessel is made up also of two different cell types, muscle on the outside, blood vessel lining cells on the inside. We put them on these bioreactors so they get exercised. On the far right, you see that a blood vessel that we implanted. This is a carotid artery. That's the artery that goes from your neck to your brain. And we were able to show that these were able to function. And it was published in that journal called Nature Medicine. All right, other tubular structures include the heart valve. This is a heart valve. Same strategy here. We have it in the bioreactor. We then coat these heart valves with the cells, and we put them in these bioreactors that you can see the heart valve leaflets opening and closing here. And again, just making sure that these heart valves know what to do before you put them in the patient. You're exercising them, if you will. Now, we have not implanted a heart valve in a patient yet. We're actually doing the critical studies with the FDA right now that will allow us to do that in the future. Okay, so far I've spoken to you about flat structures such as skin, and those are by far the simplest structures, the least complex. They're all complex, but they're the least complex. They're flat, made up mostly of one cell type. We've talked about tubular structures like blood vessels or urethras. And these structures, which are tubular, actually are slightly more complex. They're tubular, not flat. They have to remain open. They can't collapse. They have mostly 2 cell types instead of 1. So they're a second level of complexity. Hollow, non-tubular organs are a third level of complexity. That's like the stomach or the bladder. These organs are architecturally more complex. The cells are more complex. The functionality is more complex, and they have more interaction with other organs. And one such organ is the one that we targeted here that you see in this slide. What we ended up doing here is same strategy for these patients. We brought the patients in 4 to 6 weeks prior to their scheduled surgery. We took a very small piece of their tissue, less than half the size of a postage stamp. We then teased the tissue apart. The tissue is composed of 2 different cell types. We grew and expanded those 2 cell types separately outside the body. And basically the way you do that is you grow in one plate. When the plate gets full, you split it into 4 plates. When those 4 plates get full, you split it into 16 plates and so on until you have enough cells full of plates to create your construct. You then go back. We created this 3-dimensional structure in the shape of the organ using these materials we call biomaterials. We coated the inside with these lining cells. We coated the outside with these muscle cells, put it back in this oven-like device, this incubator, and we put it right back into the patient. And that's what we did experimentally, as you see in that journal that was shown in that slide. And that's what led us actually to do our human studies. And this shows one of those bladders, experimental bladders, showing you the picture of these bladders. And we published that also in this journal called The Lancet. On the far right, you see how these molds are created before they get seeded with cells. And we are now many years out from that experience. Okay, in this next video, I'm showing you how we're creating some of these solid structures, like the ear. This is an ear that we're creating for the Department of Defense. These are actually the oven-like devices that have the same conditions as the human body. We call incubators. These are digits that we are creating experimentally. You can see remember those muscle strips that we were making? So we're laying bone where you see it. We are laying cartilage in the middle, and we lay those muscle strips that I showed you to build these fingers from the bottom up. That's what the digits are, fingers. So we're building these fingers in the laboratory. Again, experimental, not for humans at this point, but just to show you how the technology works. In this next slide, I'm showing you a strategy for solid organs. This is actually a miniature liver that you're seeing on the top left. And what we have done with this miniature liver is we basically use very mild detergents to wash the cells away. So basically, this is one of the strategies we're using for solid organs. By far, the most complex organs are the solid organs. So what we're doing is we're using this strategy where we take these donor organs, like a liver, for example, that are getting discarded. These are discard organs which are not being used. And right now, they're just being thrown away. They're not being used. So instead of discarding them, we can take these organs, we can use very mild detergents to wash the cells away, and then 2 weeks later, we're left with something that looks like the organ. You can hold it like the organ, but it has no cells. But let me show you this slide again. On the top left, you see what happens with this organ. We are able to preserve that blood vessel tree, even though it has no cells. You see all that blue dye going through, and it's showing you that blood vessel tree. But the blood vessel tree is now intact, but without the cells. So what we now do is we perfuse that blood vessel tree with blood vessel lining cells. We infiltrate the parenchyma, or the rest of the glue, with liver cells, and we're able to create these miniature liver structures which are able to metabolize things that livers process, and they're able to secrete things that livers secrete. And we published that work in that journal that you see here, where we are able to create these miniature functioning liver organoids. They're only about the size of about $50. But the challenge for us now is, how do we make these larger? How do we actually implant these into patients? Okay, this next slide, this is actually another video that's showing you another strategy for creating solid organs. And this is inkjet printer, and what you just saw was inkjet cartridge, but instead of using ink, we're using cells. And we modified these printers so they would print these tissues one layer at a time. And you can see this here is a 2-chamber heart that is popping out of this printer. And about 4 to 6 hours later, the structure starts beating, as you see here. And these are the heart cells beating together. It takes about 40 minutes to print. It takes about 4 to 6 hours for this structure to actually start beating in unison. That was one of our very, very early printers. That was worked on by Tao Zhu, one of the fellows at our institute, where we started modifying just your typical desktop inkjet printer to start printing these structures. And we started printing about 10 years ago, and we did it as a way to really scale up what we were doing by hand, to scale up the production of these structures. And now we are building much more sophisticated printers, of course. We've come a long way in 10 years in terms of how sophisticated we can get in printing these structures. This is an X-ray that you see here, and in this X-ray, you're seeing the kidneys up there, right there. These are the kidneys. So basically what we're going to do is we're going to take this X-ray and we're going to use currently available software to reconstruct what these organs look like. I'm going to show you that in this next slide. This is straight from an X-ray from a patient, and this is software that allows us to really recognize what these organs look like inside the body 3-dimensionally, layer by layer. You can go down to where these organs are, and then we can isolate where these kidneys are, and then we can take that information and transfer that information through CAD printing software. As you see that information being transferred. Let me go back again. There it goes. You can see that information being transferred to the CAD printer layer by layer. 3-dimensionally, that information goes to our printer. And this is the printer right here. And what we do is we use a sacrificial polymer on the outside that holds the cells and the gel together on the inside. So it holds the gel with the cells until the cells and the gel are able to harden. And it hardens to a gummy bear consistency. And that polymer on the outside just goes away over time. And we are building these prototypes. These are currently prototypes. We're not implanting these in patients at all, but these are prototypes. But we are creating these miniature kidney structures that you see here. And this is a miniature kidney structure that was implanted experimentally on the far left. And you see it secreting yellow fluid consistent with what kidneys make. And when you look at these kidney structures, they have all the elements there that you would expect to have that actually allow these miniature kidneys to function. And we published that work on the far right, highlighted in that journal as well. So basically, again, like the livers, these are miniature kidney structures, about the size of $50. And for us, the question is, how can we keep making them larger so we can implant them into patients? We've also done other kinds of technologies using bioprinting. This is a technology we're working on for the Department of Defense, where we scan the patient's wound. We bring the patient to the suite, and we have this scanner on top of the patient's wound. So we can scan the wound, and we go back then with the same machine, we just print right on the patient. And the way that this works is it has this imaging head that images the wound and shows where the edges are, and then it knows how to lay down the cells. I'm going to show you that in this next slide. You can see the scanner going through the wound, and then once that information gets transferred to the computer, the correct layers get laid down over the wound. And that's a printer technology that we have right now. This is a prototype where we're showing you that this is printed with a gel, so the gel sticks to the patient so that the cells are protected throughout the printing process. So basically then, what I've tried to do for you today is really give you an overview of these organs. We have what we call flat structures such as skin. We have tubular structures such as blood vessels. We have hollow non-tubular organs such as bladders or stomachs, for example. We have solid organs like the liver, the kidney, the heart, and we're using many different strategies for solid organs. We've been able to implant the first 3 types in patients. The first 3 types: flat structures, tubular structures, hollow non-tubular structures in patients. We haven't been able to implant examples of these technologies in patients. But the fourth level of complexity for solid organs, we have not implanted those in patients yet. And that is we're using many different strategies to bring that to patients. We're using the strategy where we take donor organs and we wash the cells away, like we did with the liver. That's one strategy. Another strategy is to print them, like I showed you, with using these 3D printers. Another strategy is to create these cartridges that we can insert in the organs, and that's yet a third strategy. But another strategy is just to inject cells in the organ. And you can inject cells from the patient, or you can inject stem cells. And in fact, we are starting these clinical trials injecting cells, for example, for kidney therapy this coming year. Let me show you then in this slide about stem cells and what they're all about, because I'm sure that this is a topic that always comes up. First of all, for everything I've just shown you, we're using the patient's own cells. Up to this point, everything I've discussed with you tonight, we take the cells from the same patient. So the patient comes in, they have an injured tissue, we take a small sample from that patient's organ, we grow the cells from that organ, we put them right back into the same patient so there's no rejection. That's how the technology works. But let's say the patient presents and they don't have that specific organ. Let's say they were in a very bad accident and they have no organ left. Or let's say we're dealing with one of those three organs we talked about, like the liver, the pancreas, or the nerve, where you do need stem cells. What do you do then for some conditions where you do need the stem cells? Well, that's where stem cells come in. And up until recently, there were three types of stem cells that were published in the literature that could give rise to true stem cells. And there are three stem cells until recently only, what we call embryonic stem cells, which come from embryos, or the more recently discovered iPS cells, or induced pluripotent stem cells. And these are really interesting cells. Basically, these are cells that you can take from the skin or from anywhere in the body. And by using specific proteins and genes, you can revert that cell all the way back to having the very similar properties to a human embryonic stem cell. So any cell in your body can be reverted back to a true stem cell. Or bone marrow. We can take cells from your bone marrow and also use them as stem cells. So these are three stem cell populations that have been described just in the last few years. And they all have their pluses and minuses. In this next slide, you're going to see some of the pluses and minuses. Basically, when we talk about embryonic and induced pluripotent stem cells, these cells, they grow very rapidly. They double in number every 36 hours, so they're very powerful. But unfortunately, they also have the potential to form tumors. That's some of the challenges that you see with these cells. That's why you're not seeing a lot of clinical trials with these cells. They do have the potential to form tumors. And of course, they are coming from other sources, so they also have the potential to reject. On the other hand, you have adult bone marrow stem cells, and these stem cells, they will not form tumors because they're adult cells. They will not reject because you can take them from your own bone marrow and put them back in. The bone marrow is the stuff that's inside your bone. We call it bone marrow. But unfortunately, with these cells, you cannot necessarily get these cells to grow outside the body. So on the one hand, we have stem cells that you can grow, but they can form tumors or they can reject. On the other hand, you have stem cells that will not form tumors, will not reject, but you can't necessarily grow. So approximately 10 years ago, we started looking at an alternate source of stem cells. And we started looking at the amniotic fluid. That's the fluid that surrounds the developing baby in the womb. And also the placenta, which is also called the afterbirth. Right? So when the baby's about to be delivered, the first thing that happens is the water breaks. That's the amniotic fluid. Then the baby gets delivered, and then comes the afterbirth. The afterbirth is the placenta. So it turns out that we postulated 10 years ago that maybe, just maybe, both the fluid and the placenta had a stem cell population, because we knew that both the fluid and the placenta had cells from the developing fetus. The question was, could we isolate a true stem cell? And that's exactly what we did. In this next slide, we show you that we collected fluid and placenta from over 400 pregnancies, and we were able to, in fact, to find this stem cell that we were able to drive into all three major layers that give rise to every tissue in your body. And we showed that in this initial study that we could get these cells to go into many different directions, including bone, fat, muscle, including heart, the cells that line blood vessels, liver cells, cartilage, and other cell types. But basically, these cells are unique. These are a new class of cells that we described that are neither embryonic nor adult, but they have properties of both. The cells grow in large numbers. They double in number every 36 hours, just like human embryonic and iPS cells. They double rapidly, but interestingly, they don't form the tumors. And you can save them at the time of birth for self-use, so you can also avoid rejection. No cell is perfect, and neither is this one. And the weakness with this cell is that for these cells, it takes a little bit more work to get them to go to where you need them to go. So, for example, if you're talking about human embryonic or iPS cells, these cells will turn into a liver cell within 7 to 10 days. For these cells, it takes you about 4 weeks to get them to a liver cell. So you have to work harder to get them there. But that's also the same property that does not allow these cells to form the tumors, because the cells are slightly more developed than the human embryonic stem cells. They're not as primitive, so they will not form the tumors. And we highlighted this work in this journal, Stem Cells from the Womb. So basically what we have now is we have what's considered embryonic stem cells. We have what's considered adult stem cells, and this would fall under the category of perinatal cells, which is also in the same category as other cell types like cord blood, etc., with the difference that these cells do expand in large numbers and do form other tissue types. So basically then, what I've tried to do for you today is really give you an overview of how we work in this field that we call regenerative medicine. And as I mentioned, we have many cells that we use, but we rather use a patient's own cells, because these cells are the patient's own cells. They're tissue-specific cells. Like if we want to create a windpipe, we're going to take cells from the windpipe. Those cells already know they're a windpipe cell. We don't need to use a stem cell and transform it. These cells also will not reject. Many people ask, what's the best cell? There is no best cell. It really depends on how the patient presents. But the best cell at the end of the day is the patient's own cells that will not reject and will not form tumors. One of the major challenges I have when I give an overview of this type is that, here I'm giving you this 40-minute overview, and you're going to walk away from here thinking, boy, isn't that stuff easy? You just put some cells on a scaffold, and there comes the organ, Well, nothing could be further from the truth. Actually, these are very, very hard technologies. Very hard technologies. And it really takes a multidisciplinary team to get these technologies to work long term. So these technologies do require, as I'm going to show you in this slide here, requires growth factor biologists. It requires cell biologists, material scientists, bioengineers, biochemists, physiologists, pharmacologists, physicians, all working together to bring these technologies from the bench to the bedside. So it really takes a lot of work and a lot of talent. But when it comes to research, really, we do follow method one. We really do follow method one. And what that really means is that we really do things very methodically. When you're doing the research, you cannot afford to do anything but be methodical, which means we make sure that we do all the basic biology of the cells before we ever even match it with materials, that we pick the right materials, that we do the right studies, that we replicate the studies, and we do them over and over and over again till we know for sure these technologies are going to work. And this team that we have, this multidisciplinary team, we basically bring everybody together, and once we feel like we have this technology down, that to the point that we can take it to the patient, we actually bring the whole team together, and we ask ourselves a very important question, which is, are you willing to put this in your own loved one? Are you willing to put this technology in your own child, in your own parent, and your own spouse. And unless we all can agree to move forward, we will not move forward unless we all agree to do so. Unless we all feel confident that we can use these technologies in our own loved ones. And when we implanted these technologies in patients, we did so very carefully, very slowly, and very methodically. Because at the end of the day, we wanted to make sure that we observed our oath for medicine, which is primum non nocere, first do no harm, right? In Latin, first do no harm. That's really the oath that physicians take, and it's very important that with these technologies we do no harm. This is a historical highlight that you'll see in the slide that was in Science, the journal Science. And it shows a history of technologies. And I just want to point out one technology here, which is this right here. This is an iron lung right here, an iron lung. How many of you remember when the iron lung came out? Please raise your hands. Wow, keep your hands raised. Look around you, everyone, because, okay, you just dated yourselves. But basically, when the iron lung came out, this was considered one of the biggest advances in medicine when it came out. And the only place you see an iron lung now is in a museum, right? And 60 years later, we look back upon this technology and we say, boy, wasn't that primitive. 40 to 50 years later, we look back at this technology and say, boy, wasn't that primitive. And here on the top right, you see that one of these engineered organs. I can assure you that 60 years from now, people will look at these technologies and say, boy, wasn't that primitive. Because our goal, and the goal of our institute, and this is what I've charged our the young generation of scientists, is that our job is really to make our own technologies obsolete. To make sure we keep pushing the envelope and make sure that we keep improving these technologies to get them to our patients and make them benefit from these technologies. At this time, I'd like to acknowledge some of the funding sources that have made this work possible for us. And finally, I would like to leave you with a very short clip. This is a video clip of a patient who was interviewed 3 years ago. And he had one of these organs implanted, so I'll let you hear this.
I was really sick. I could barely get out of bed. I was missing school. It was just pretty much miserable. I couldn't, go out and play, basketball at recess without feeling like I was going to pass out when I got back inside. I felt so sick. I was facing basically a lifetime of dialysis, and I don't even like to think about what my life would be like if I was on that. So after the surgery, life got a lot better for me. I was able to do more things. I was able to wrestle in high school. I became the captain of the team, and that was great. I was able to be, the normal kid with my friends, and because they use my own cells to, build this bladder, it's gonna be with me. I got it for life, so I'm all set.
That is Lucas Masella. He's now 13 years out from having received his engineered organ. We still have many challenges ahead. I had the pleasure of seeing Lucas after 10 years, 3 years ago, for the very first time after he had his surgery. When I think of Lucas, I know that even though we're able to place these organs into patients, we still have a long way to go. We have a lot of challenges in terms of scale-up. We have challenges in terms of cost. We have regulatory challenges that we still have to overcome. There are many challenges ahead. But one thing is certain, and that is that these technologies do have the potential to make patients' lives better. And for us, that's the promise of regenerative medicine. It's not about the cells we use or the technologies we choose. It's all about making our patients better. Thank you for your attention tonight.
we've got some time this evening for questions, and what I'd like to ask people to do is if you have a question, if you could step forward to the mic. That means that not only everyone in the room will hear you, but also anyone who's watching this at a distance will have a better sense of your question. So we've got mics in the center aisle there, so please, someone take the plunge and step forward and ask the first question. And there may well be a prize.
Hi, I've really enjoyed your lecture and I've learned a lot. One of the tissues that you regenerate on your list or from your list is the nerve cells. Our province has a very high incidence of MS, and I'm wondering if you see any future for helping patients with MS using regenerative medicine.
Yes, so as you know, MS is actually a central nervous system problem. So it's a widespread challenge that occurs in the nerves of the body. And so there are strategies right now to treat MS with stem cells, and there's a number of trials now ongoing actually in that area. And the trials involve the use of bone marrow stem cells, which have very special immunomodulatory effects, or a better way to say that is they can deal with inflammation in the body. They have ways to control inflammation in the body. There's no data out yet in terms of the true outcome of these trials, but hopefully those will be coming out soon.
I'm kind of an amateur, but I'll take a stab at this. Are we doing something at the DNA level with these cells once they're put onto these systems like zinc finger nucleotides, altering things, sequences, and trying to get them to grow in a state that's more productive?
So, we are not manipulating the cells at all at this point. We basically just take the primary cells, just grow the cells, and put them right back in. Now, we do have a technology for muscular dystrophy that came out of the Institute, and that's actually geared to go towards clinical trials in about 18 months or so. And that is a technology where, as you know, with muscular dystrophy, every cell in your body has that genetic defect, where the muscle cells don't have that gene. So one of the things that was done was basically taking the muscular dystrophy gene, and so we take a small biopsy of the cells, muscle cells. We grow and expand the cells outside the body in large quantities, but then insert the gene that's missing and put those cells back in. And that's experimentally worked nicely, and now that's being produced for clinical application in the near future.
Do you see this as a computational issue in the future that could help greatly, where synthetic biology and those sorts of things can aid you in creating these things?
Well, it's interesting. We as you know, what we really want to do is we just want to preserve the normalcy of the cells. I mean, that's because any time that you start messing with the actual makeup of the cells, it becomes very problematic for the regulatory bodies and to put into patients. You just open up a Pandora's box in terms of problems. So certainly for experimentation, for experiments, I think, things are being done right now to see how you can enhance cell functionality and how you can make cells better. But at the end of the day, our goal is really to take the patient's own cells and give them back the organ the way they had it before. Unless you have single genetic diseases like muscular dystrophy or cystic fibrosis, where you want to put the gene back in. And as you know, there are mechanisms to do that with gene therapy, but the question is, how can you combine it using these cell therapies? So to answer your question, there's a lot going on in the experimental phase, but nothing that would be safe currently that you would want to transfer to humans.
Thank you.
Good evening. Are you doing any work in conjunction with blood, specifically where in solutions to leukemia or lymphoma? That type of thing?
Yes. So right now, as you know, the therapies for leukemia or lymphoma, for example, involve That's where hematopoietic cells come from bone marrow. From bone marrow. So that's where bone marrow banks. You know, I'm sure you've heard of bone marrow banks or cord blood banks. And these cells, bone marrow and cord blood, are very, very specific to forming cells that actually can treat leukemias and lymphomas. But the challenge with both of them, as I mentioned on that stem cell slide, is that you really cannot get these cells to grow outside the body in very large quantities. So anytime you take a unit out of a bank, a bone marrow unit out of a bank, or a cord blood unit out of a bank, you have to replenish it because these cells will not divide. And so the way to get one way to get around that is to use these cells I just mentioned, the amniotic fluid and placental stem cells. We published that initial work back in 2007, and there are many papers out in the literature right now with many different scientists working on these cells. But probably the most exciting paper I've seen to date is one paper that came from the University of Paris and the Pasteur Institute, where they took these amniotic fluid and placental stem cells, and they injected these cells in a mouse model that was bone marrow depleted. So it didn't have this mouse model did not have a bone marrow, and when they injected these cells, it was able to replenish the bone marrow in these animals. And the reason why I find that exciting is because you now have a system where you could possibly have a universal bank that once you save the bank, you never have to replenish it again. Because you can just keep taking these units, you can just keep expanding the cells outside the body without actually having to take bone marrow from other patients to do so. So yes, we are looking at that therapy using these amniotic fluid and placental stem cells I discussed with you earlier.
Thank you.
Well, good evening, Dr. Atella. I really appreciate your lecture here, and I have a question about the kidney you showed in your video. Like the engineered artificial kidney is right now functional, or just you said you use the biomaterial to make the shape and use the cells and gels to fill the material to let the cells regenerate to form the kidney. So it's right now can be functional or just to make the shape.
Yeah, actually, so it's exactly what I meant, what I showed. That actually the printed kidneys we have not implanted yet. Mm-hmm. They're just prototypes. But we do have the miniature kidneys that we have implanted. In other words, those large kidneys that you saw, the prototypes, we have not implanted yet. But we have implanted the miniature kidneys that you saw that are able to actually make urine. And we implanted those into cows and steer. And we were able to show that they could actually, make these kidney-formed structures that were making urine and secreting the things that kidneys secrete, like erythropoietin and vitamin D3, so they can do that. But we have not made large kidneys that we can put in a patient, no.
Well, so what's the obstacle here to make it for clinical test?
Well, it's the challenge is so, we've implanted flat tubular and hollow non-tubular structures in patients, but not solid organs. And the reason for that is the vascularity. Really, you need massive amounts of nutrition and vascularity to make these organs survive long term. And so we're working on that, but it's not yet ready for patients. We have to be absolutely sure that these technologies are safe and reproducible before they get implanted in patients. Of course, we've done so experimentally, like in cows and steers, but not in patients. Now, for kidneys, as I mentioned, we do have some of the strategies, like the 5 strategies that I mentioned, and so we are using cells for therapy. So we're starting a clinical trial this coming year, for patients with kidney failure that are going to be treated with cell therapy, which will be the first venue for therapy. But we do not feel at this point that the technology is ready for prime time yet to implant a solid organ.
Okay, thank you for your answer. And one more small question is that, so when you do the the artificial kidney implant into the patient, the biomaterial already being dissolved or disappeared, so it's already a total artificial kidney, or the biomaterial which support the cell is still there and it will degrade it in the patient body.
Yeah, so all the materials that we use, every single one of them degrades inside the body. So the only thing that stays behind are the cells, and the cells lay down their own material. So the way that it works is that as the cells are able to, as the material that we put in goes away, the cells are able to lay down their own glue, if you will, that keeps the cells together. And that's how the technology works for all the tissues we've implanted.
Thank you.
Thank you very much for the lecture. It was fascinating. I'm curious, one of the organs you didn't mention was the brain, which is probably one of the most complex. I'm curious if you have any thoughts on how regenerative medicine might work for, say, either genetic types of things like maybe neurological disorders like dyslexia, or with disease, maybe Parkinson's, or even brain injuries of one type or another, if there's ways to, if you see any future for regenerating to deal with those kinds of things.
We do. We do see a future when it comes to cell therapy. So same challenge, for these organs. Brains are not being engineered right now, for example, or miniature brains, or in terms of putting them in animals or anything of the sort. The bottom line is that when it comes to the nervous system, it's a very complex system. So cell therapies are being now used for traumatic brain injury, and they're being used clinically for traumatic brain injury. We'll see what the clinical trials show at the end of the day. But there are many different cell types that are being used, but these are the kind of things that are also being used for Parkinson's. So conditions such as Parkinson's and traumatic brain injury, which you mentioned, Bob, they're all really being targeted right now with cell therapies. And so, but we don't have the outcomes yet. I mean, experimentally, they look promising, but we'll see what happens long term, because cell therapies are not as easy as they may sound, because you really do have to make sure that the cells integrate and that they do what they're supposed to do. So most of the cell therapies being used today are being used more like immunomodulatory, in other words, controlling inflammation. But they do seem to have very good outcomes, and I know for a fact that you're going to see some interesting papers coming out, showing that they do have benefit for brain injury. So I think the future looks bright, but in terms of engineering structures and putting them in, that's not in the near future, or certainly not in the next few decades anyhow.
Okay, thanks.
Dr. Atala, that was a beautiful lecture. I really enjoyed it. You showed success or clinical success in the bladder reconstruction in that young man, and the demand worldwide for that is huge, for bladder reconstruction or bladder replacement. I just wonder, has that been sort of transferred into other institutions, the procedure or the technology, or it looks like a huge amount of resources are required for that, and I just wondered how, clinically useful, or in what sort of a magnitude is being employed right now for bladder reconstruction.
Yeah, so it's still under clinical trials. It was really the first organ that we implanted, and because of that, we really had to wait a long time, for before we went to a phase 2 trial. So we really wait. In fact, we waited over 8 years after we implanted the first one to go into a phase 2 clinical trial. And so, and people asked us, well, why did you wait so long? Why? Because we really didn't want to rush, and we had no idea what these organs were going to do long term. We had absolutely no idea how they were going to react. So of course now we are, more than 13 years out, 14 years out from having implanted them. So now we can start accelerating the process We're entering phase 3 trial pretty soon with the bladders, and they have been done in several centers around the U.S. We've learned a lot in the process, and what patients to treat, what patients not to treat. For example, we learned that, in the phase 2 clinical trials, we learned that you really do need to make sure these bladders are able to cycle. That is, you don't want to put it into patients that don't have the ability to cycle their bladders. That is, letting them fill and empty, which makes intuitive sense, right? Because if you're not giving the bladder that signal, it's not going to develop normally. So things that we learned from the phase 2 clinical trials is that patient selection is very important. You have to make sure that you give the organs the right cues that they're supposed to have. And we're going to be starting phase 3 in the near future. So there are about 10 centers around the U.S. that have been participating that participate in the phase 2, and we hope to increase that in the future as well.
Good evening. I was wondering if anybody in this field is looking at electromagnetic rhythms to support, well, actually there's a few areas where I could see it be helpful.
Yeah, interestingly, so electromagnetic fields and energy has been used. Is that you're talking about the energy, electromagnetic energy?
The rhythms.
Yeah, so that's actually been used and has been shown. We've actually used it in our laboratories to see how cells are able to react. And in fact, there is an improvement in cell growth when you apply the electromagnetic fields. There's an improvement in cell growth. Now, interestingly, it's not that relevant eventually, because the cells will eventually grow, right? But you are accelerating their development initially.
Time will be the essence in some cases, I assume.
I'm sorry?
Time will be an essence in some cases?
Because the cells grow exponentially, very rapidly, at this point it doesn't give it an advantage, but you never know, it may in the future.
Yeah, functioning, or because you still have the liver and the pancreas, and nerve cells are complicated, but they're definitely in rhythms all the time.
Yeah, I think there's a definite positive effect. Nothing at this moment that we can say it's a major advantage, but they do better.
Good luck.
No doubt about that.
Good luck.
Thank you.
Thank you, Dr. Atala, for your lecture. I'm glad you like Saskatoon. As you know, macular degeneration is one of the common causes of irreversible vision loss in the developed world. I was wondering if you think your printing technology can be used to print retina or layers of the retina for treatment of that disease. I've read in the literature that stem cells have been inserted into the eye with some success, with return of vision for that problem.
Yes. So now bioprinting is probably it's not going to be the first indication. The retina, what's really nice about the retina is that it's a very small space. So you can actually, it really lends itself nicely to cell therapy instead of engineered tissues. So there are currently several trials out there right now where stem cells are being injected into the retina, like for Stargardt's disease, with really interesting outcomes. Actually, I think that's going to be a nice paper when it comes out. So I think that unless you have a totally shattered, retina, an engineered retina will not make sense. That would be highly unusual, though. So it's probably not a first indication for our bioprinting technology. So for the bioprinting technologies, we intend to follow the same strategy that we did for other organs, in terms of trying to achieve flat structures first with bioprinting, then tubular structures with bioprinting, then tubularized, then hollow non-tubularized structures, and finally, hopefully, the printed organs, the solid organs. But we plan to follow the same strategy that we have with the handmade organs with the bioprinting.
What do you think about, like, for globe rupture or situations like that where most of the retina is shattered? Do you think it would be possible for situations such as that?
Yeah, I think so. That's a very rare. That would be extremely rare, though. It's not a very common condition. Usually what happens is you may have a retinal detachment that can be, or you may have Stargardt's disease or certain diseases of the retina. It would be highly unusual to have an entirely shattered retina. But if that were the case, and and we were at that stage, certainly, I don't see why it couldn't be done. I just don't think it'll be one of the first indications for it.
I have a quick question in regards to the hair cells regeneration into the cochlea, and how far is it?
Yeah, so it's interesting. We are actually working on hair regeneration in the cochlea, and we've basically have done studies showing that we can induce hair regeneration in mice, in rodent models, and we are working very hard to see whether we can keep translating that up the ladder. We published a paper recently in that area, and we continue working in that area, trying to really induce hair regeneration. As you know, for those of you who, the cochlear hairs are the hairs that are inside the ear, and the problem is that if you lose those hairs, you go deaf. I mean, that's the bottom line. And those hairs don't grow in adults or in humans. They just don't grow, apparently. Or certainly that's what's the dogma, that they don't grow. So what we are trying to do is trying to figure out, how we can make these cells grow. And of course, there are many groups around the world trying to do the same thing. We're just one group of many. But we just published a paper recently in that area, and we keep working at it. That's going to be a long-term project, though, I'm sure.
Wonderful. I think we have time for one more question.
Good evening. I was just wondering, has there been progress made in decreasing the invasiveness of these procedures?
Yes, there have been, and I think that's really unrelated to the Regen Med space. It really has to do more with the advances in minimally invasive surgery and robotic surgery. It's just amazing what's happened in the last decade alone with robotic surgery, and how now surgeries can be done with very small incisions, and patients can go home the next day. And a lot of the stuff that's going on with actually introducing surgeries with invasive imaging technologies, through catheters. So it's interesting, when I'm sure most of you were used to open heart surgery, right? If you had a blood vessel. But that procedure is seldom ever done now. Now, I grew up with that procedure. When I was trained in surgery, we used to, open heart surgeries were being done. We had a whole intensive care unit full of open heart surgery patients. And now, most of those patients are being treated with stents. They come into the hospital with a heart attack. They get taken to this X-ray suite, and they put a little stent that pops a blood vessel open. So it's just amazing to see how minimally invasive techniques are really taking over surgically. Unrelated to regenerative medicine, but of course regenerative medicine will take advantage of those techniques, and also minimally invasive surgery will take advantage of regenerative medicine techniques because of the way that you can deliver cells to these sites of injury.
Thank you.
Just a quick word of thanks to the Bessborough for hosting us this evening, to the media access and production people who you see around you working all the technical angle for us. As well, Dr. Atala mentioned at the beginning of the evening that thanks go certainly to Vanessa and to Vanessa Ross-Young and to Karen Hayward from the Center for Continuing and Distance Education. Karen would insist that I give most of the credit to Vanessa for all of her hard work in organizing. We do have a reception to follow the lecture. We invite all of you to stay. Some of you may have been a little reticent to come to the mic to ask a question, and there will likely be opportunities to ask questions at the reception. And now, finally, if I could just offer a word of thanks to our Whelen Lecturer, Anthony Atala. I think your work, I think the audience will agree with me that your work in the science and technology of regenerative medicine is an inspiration. While it may well be that 60 years from now, we've decided that this is obsolete, tonight, The cool factor on this work is off the charts. Your capacity to share your work, as you did tonight with us, in such a way so that it's accessible and well understood by all, is certainly a gift. And we thank you for inspiring us at the University of Saskatchewan and our community members. I hope you'll leave knowing that you have a fan base here in Saskatoon and that we would welcome you back at any time. And we offer you a token of our thanks, a small gift.